Every year, lot of people are dying throughout the world. Out of this death, total death throughout the world, 1/3 of the death are due to coronary heart disease. And its prevalence is gradually rising, particularly in developing countries like India. Here the prevalence is higher than any other country. Most of the burden of cardiovascular patient globally, out of total burden, the maximum belongs to India. Why? This is rising. This is rising because of two important factors. One is increase in life expectancy, or why it is increased? Development in health sector. Treatments are available and the services are available. So this is one reason. Another reason is, which is very threatening, is our lifestyle. Improper lifestyle we are adopting. What are the improper lifestyle? Sedentary lifestyle. And improper diet. Fatty diet. We like to have fatty diet, spicy diet, smoking, alcohol consumption, sedentary lifestyle. And nowadays, the amount of, out of 24 hours, we are devoting our time into this. So when we use this, we cannot do any physical activity. So we remain. Because of this, we are spending much of the time sedentarily. So all these are important factors for development of cardiovascular diseases. So our lifestyle is very important factor. Moreover, the present lifestyle is full of stress. See how much stress you are facing. You are young, that's why you are able to adapt it. You are leading an anxiety-full life. Lot of stress, anxiety, depression. So these are also psychological factors that initiate, that develop cardiovascular, help in developing cardiovascular diseases. Coronary arterial diseases. So, we must, since you are going to be taking care of such patients, so you must have proper knowledge, adequate knowledge about this coronary heart diseases. In the heart, we have different types of, the heart may have different types of disease. We may broadly classify into 3. One are because of abnormality in the myocardium. So this myocardium is always, since we developed our heart, from that day, and we will be continuing it till death. So it has to work continuously, without any pause, without any rest. So lot of work it has to perform. And for this working of the myocardium, every work needs energy. And this energy is supplied by the nutrients. And only nutrients will not do, they are to be oxidized, they are to be burnt. So for this purpose, we need oxygen. So oxygen and nutrients has to be delivered continuously to the myocardium. If there is any problem in supplying the oxygen and nutrients to the muscles, myocardium, then there will develop myocardial disease like myocardial ischemia, myocardial infarction. So that will develop. Another type of disease of the heart is valvular disease. We know there are AV valves. These AV valves are tricuspid valves and bicuspid valves or mitral valves. Bicuspid valves are also called bicuspid valves. And these two valves are having special arrangement. They are having some tendinous structure, chordae tendineae. So disease may occur due to tearing of the chordae tendineae. In that case, the valves will be bulge out and there will be opening. So ventricular blood, when ventricular will be contracting, their blood, instead of going to the vessel, it will be entering again into the atria. So this is another problem. So this sort of disease is a valvular disease. The Purkinje cells, not Purkinje cells, these are, what are these? The tendons, chordae tendineae remains attached to the muscle projection of the ventricle. Inside there is some projection. So with this they remain attached. So these muscles can also become paralyzed. Same thing will happen. Result is same. There will be bulging. Okay. So this paralysis of these muscles can also lead to valvular disease. Also there may be sclerosis. Sclerosis, deposition, the valves may become heavy. Why? Because of deposition of calcium. The valves will become hard. As a result, they will not, when they will be closing, they will not be properly closed. So there will be some opening, so that through that opening, either blood from the aorta, they will be coming back into the ventricle again. But normally it doesn't happen. Normally from pulmonary artery and from the aorta, they don't come back because of the closure of semilunar valves. So if there is defect in the semilunar valves, they may come back. There may be regurgitation that you can diagnose hearing the mama. Okay, so this is another type of disease. That is sclerosis of the valves. That is, in short, there are valvular disease. There are myocardial disease. And another disease of the heart is another group of disease that is due to, see, our heart is having a generator. It produces current intermittently. Okay, and this generator resides in AC node. We'll take another class in our lecture theatre. I will take the next class on this. So we are having conducting system in our heart. These conducting systems are specialized cardiac cells, modified cardiac cells. Their action, their function is not to contract, relax. Their function is to carry or generate cardiac impulse and to carry this to the auricular muscles or to the ventricular muscles, like electrical waves. Okay, so if there is any, there can happen. The AC node is producing impulse. That impulse has to be spreaded throughout the atrium. Then it has to come into the AB node. From AB node, it has to travel through the bundle of his. Then from here to the right bundle branch, left bundle branch, and then Purkinje fibers, then to the whole ventricular muscles. Then only ventricular muscle will get stimulation and they will be contracting. So if there may become develop certain blocks. It may be in the right bundle branch, it may be in the left bundle branch, it may be in the fascicles, it may be the SA node, AB node may become dissociated. So these are the conduction defects. We heard left bundle branch, so somebody is suffering from LVBB. They require, heart is not pumping or beating regularly, rhythmically. There is irregularity. Yeah. There is irregularity in heartbeat. So this is because of conduction defect. This is because of conduction. In that case, for our survival, we need pacemaker. Such as blockage. When there is pacemaker is needed, when there is blockage in the defect in the conduction system. Okay, so this is the remedy. But there may be blockage in the coronary circulation. And this blockage is different from this. We will discuss it. So now, today, we will be learning about coronary circulation. We will be learning about coronary circulation because this problem in the coronary circulation give rise to disease like ischemia and myocardial infarction. So let us first study what is coronary circulation and what is their course, where from they are originated and how they branches and where these branches are going. Okay, ultimately how they are reaching to myocardium. So this is the first thing, the anatomical structure of the course of coronary circulation. Okay, so this coronary circulation is having arterial system as well as venous system. So first we will focus on the arterial system. The arterial system, these are different from systemic arteries. Here, the arterial system, there are two main arteries, left coronary artery and right coronary artery. Both the artery originate from the root of the aorta. One aortic valve is there, just after the valve, at the root of the aorta, there are valves, semilunar valves. Just over the valves, when the valves open, so it consists arterial supply, venous supply, and lymphatic drainage. Okay, we'll discuss one by one. The arterial supply, the cardiac muscle is supplied by two coronary arteries, the right and left coronary arteries, and both arteries arises from the second point. Both the arteries arises from the sinus behind the cusp of the aortic valves at the root of the aorta. So this is their origin. Again you go through it. First I will show you the diagram. Okay. This is the aorta. This is the root of the aorta. Aorta origin, this is the wall of the atrium. Wall of the atrium. So this is the root. And this is the outlet of the ventricles. Okay. So see, here are the semilunar valves. Actually, this is the artery. I have slitted it longitudinally. Then I make it flat. So now it looks like this. Now you see there are three cups. Okay, and the cups, you can insert your finger within the cup, and this must, if you try to insert your finger, so this much it will enter within the cup. So there is a sinus, cavity-like structure within the cavity. Okay, and these valves remain attached to a ring-like structures. And this is the fibrous structure. So within this ring, the cusp remain attached. These ends, the other ends remain attached. And these are the free ends. of the cusp. So when the blood tries to enter within the ventricle, so they close. They come closer, then they close. Okay, and they will produce one sound. And this is the second heart sound. Remember this. This is the second heart sound. When suddenly, because of pressure, backflow, there will be ventricular relaxing. Okay, so what will be there? So blood that has entered into the aorta, they will be trying to come back within the ventricles. So when they are trying to come back, so these valves will close. So they will this blood will be unable to enter within the ventricle, and it will produce a sound. This is the which sound? Second heart sound. Okay. Now see what is there at the back of this. This is the wall of the aorta. This is the wall of the aorta. See, there is a hole. There is a hole here also, but there is no hole here. Okay? So this leaflet, and at the back of the tube there are opening of left and right coronary artery. So from here, left and right coronary artery arise. Now one thing you will see, when ventricle will contract, they will push the blood towards the aorta. Now what will happen? This leaflet, they will be opening. They will be opening. And they will be, these cups will be covering the wall, this part. As a result, less blood will be entering because the leaflets, the cups, they will be obstructing the flow of blood during systole. During systole, when blood is entering to the aorta, the valves are opening. Okay. Suppose this is the hole from which blood will enter. So blood is trying to. So now what is happening? This is opening. So as a result, these are covered. The holes are covered. So sufficient blood cannot enter within the, during systole, you will see there is biphasic coronary blood flow. I will explain it. So this is one reason. During systole, less blood enters into the coronary cell. But in other organs, we get more blood during systole. But here you will see, particularly in the left ventricle, less blood is flowed in the myocardium. This is one reason. Another reason I will explain. Another main reason I will explain. This is not the main reason, but this is part of the reason. And take care. Come here. Be attentive. Come here. Sit here. And now see, look at this picture. These are the mitral valve, bicuspid valve, and this is the tricuspid valve. When it opens, the blood will enter into the pulmonary arteries and go to, this blood will go to lung. And when they will be opening, when they will, I'm very sorry. From atria, the blood will enter through this valve into the ventricle, and from left atrium, blood will enter through this into the ventricle. Okay, and see, this is the base of the aorta. This is the base of the aorta, and this is one cardiac, this is one coronary artery coming out, this is another coronary artery coming out. Okay. So both arteries arise from the sinus. From the sinus, that is the trunk or root of the aorta. So there are three cusps. One cusp is not having any. At the back of this cusp, there is no coronary artery. Only left and right cusp is having the coronary arteries. Is it clear? This picture is clear to you? See. Now, let us discuss how about the first right coronary artery. This is right coronary artery. Okay? Right coronary artery. And this is left coronary artery. This is arising. This is what? This part? Right atrium. This is the auricle. This is the auricle of this. So in between, this is the aorta, base of the aorta. So in between these two, auricle and pulmonary trunk, from the wall, this has originated. This is the base of the aorta, root of the aorta, from here it has originated. Now it will pass along the coronary sulcus. You know the coronary sulcus we have seen? You have seen it? In anatomy class, you have seen it. So this is a groove. So through this groove, the right coronary artery will come out. Will come and they will giving out the branches and they will go back. And it will go back. Let us see how they produce branches. This is what? Right coronary artery. This is one branch. What is this? Sinoatrial nodal branch. That means this branch supplies blood to the SA node. This branch supplies blood to the SA node. And then this. What is this? Right conus. At the base there is pulmonary trunk. At the base this area is trunk conus area. So they will give one branch to this area. And the second branch, where it is going? To the conus area. Okay, what is the name of this branch? Right conus. Okay, then it will give branch to the atria. This branch will supply blood to the atrium. Mostly, which atria? Right atria. And it may supply the part of this, the left atria also may get some blood from this branch. Okay. Then, what is this? Right anterior ventricular branch. There are two ventricles, right and left ventricles. So in between there is a group. What is this called? Anterior interventricular sulcus. Sulcus. Group. What? Sulcus.
It is groove, so it can be called sulcus.
Yes, correct. This dotted means it has gone to the back. It has gone to the posterior side. Now it will travel, it will be bending towards the posterior aspect. So there it will produce branch. And what is this branch? This is the posterior descending artery or posterior interventricular artery. Means this branch will travel along the group. Posterior side also having one subgroup. So along the group it will be descending down. Okay. So these are the different branches the right coronary artery gives off. Now let us see where they supplies blood. So these are the course. First branch to SA node, then branch to right conus, then branch to atria, then right anterior ventricular artery, then right marginal artery, and then right coronary artery turns back and reaches to posterior coronary sulcus, and then descends down along posterior interventricular sulcus, forming PDA. PDA means posterior descending artery. Posterior, when an angiogram is performed, angiogram means this angiogram. One catheter is inserted, and this will be sent to the heart, to the aorta. Through aorta, it will be coming to the root of the aorta, and the catheter will enter into the right coronary artery. And when at the opening of the right coronary artery, they will inject some dye, radio-opaque dye. Now this dye, along with blood, this dye will be circulating through this. When there is any blockage, this dye will not be passing next to this area. Then, in this way, this blockage is identified. Where the block, in which branch it is. Sometimes, in most of the cases, we get in PDA. In PDA, what is PDA? Posterior descending artery. So this is important. And in the report, PDA intact, or obstruction is there, blockage is there in PDA. So this sort of repeat. What is PDA? You should learn. Or you can get also LAD. Left anterior descending. Anterior. In between two ventricles, there is one sulcus. Through this one artery will be descending, and that is the PDA. So two sulcus, anterior, posterior. So there will be two main arteries will be coming down, and they may have, these two arteries may suffer blockage, and this can be identified using angiography, and this can also be removed by angioplasty. So what is angioplasty? How it is blockage? Simultaneously, when they find out the blockage, then they will through the catheter, within the catheter, at the tip of the catheter, there is balloon. So they will reduce to the blockage, and gradually they will inflate the balloon at the tip. So because of inflation, the artery which is narrowed, it will be dilated slightly. Again, they will insert it further. Again, the balloon will be dilated. Okay, done. Some part is dilated. Again, deflated it, proceeded further. Again, inflated it, dilated. Again, deflated and proceeded further. In this way, this narrow area becomes wider. Now, if it is removed, again it may become constricted. So what can be done? They insert one spring. This spring will be there, staying there permanently. Now it cannot constrict further. So in this way, block can be removed. And previously it was the blockage is here. This is the line. Blockage is here. So from somewhere a piece of artery is taken and it is joined this part to the bypassing. We have bypass road, no? So this sort of bypass road they made. These arteries, the main you see, you will be able to see this artery because they are located in the epicardium. They are located superficially. What is the three layers of wall of heart? Outer, inner myocardium, in between, and the innermost layer is endocardium. Okay, so the bigger arteries means left and right coronary artery, they will pass through the epicardium. And from the epicardium, from these small vessels, small arteries will arise, and they will be inserting within the myocardial show. So these are the branches provided by the anterior segment, and these are the branches provided by the posterior part. So see, septal, in between the septum, where in between the two ventricles there is one septum. So there are four septum, there is also branch, septal branch. Okay, AV node is there. We saw one branch is going to SA node, another branch is coming to AV node also. Okay, there is from the right bundle, from the right corner, left coronary artery, it will supply to bundle of his or right bundle branch. This is what? Here is the group. One ventricle, this is another ventricle. From anteriorly, which part is more visible? Left ventricle or right ventricle? If you see my heart anteriorly, which ventricle will be mostly visible to you? Right ventricle. And the left ventricle is mostly located in the posterior to me, posterior to the posterior side. Okay. So this branch is going back. This branch is going back, and this branch is descending down to the interventricular group sulcus. Let us see what are their branches. And this left coronary artery is larger than the right coronary artery. It originates from the left posterior aortic, same. The right side theke, now it will be originating from the left side. The trunk of artery appears between the pulmonary trunk and left auricle, where it divides into two branches. Anterior interventricular artery, which is descending down, and the circumflex artery bending and going to the backward. So it is the circumflex artery branch. Sometimes blockage happens here. It may happen here. So this is the branching of left coronary artery. See the different branches. Now, the LCA, the branches, they will be supplying different parts of the heart. What they supply? These are the portions of the heart supplied by the left coronary artery. I think the course of coronary artery, coronary circulation is most important in anatomy. Here in physiology, we will not give a most emphasis on this course of circulation. We will be emphasizing on the peculiarities, specialities of coronary circulations. And this coronary circulation are having auto-regulation. When there is, by some means, if it is flow is reduced, it will automatically, it has the mechanism by which it will increase the flow. If there is any mechanism, there is increased flow, that means it will try to bring or it will try to control the flow within a range. What is this range? 60 millimeter of mercury pressure to 150 to 160 millimeter arterial blood pressure. Okay, within this range, it will melt. If it comes below 60, then by its mechanism, it will increase it. If it goes beyond 160, the flow will be increased by it. It has a mechanism. I will explain this. What is this mechanism? There are two mechanisms. One is myogenic. There is one theory called myogenic theory, and another mechanism is there, metabolites theory. The metabolites which will be produced and they will cause vasodilatation. So we will explain this, how it happens, autoregulation, how it happens. So these are the portions supplied by the left coronary artery. Is that okay? The main, again I am repeating, the main arteries, the bigger arteries of coronary circulation, they remain superficially, means in the epicardial layer. Now they will produce branches. These branches will enter within the myocardium, then to the endocardium. We'll see next. And this is the venous system. So you learn it thoroughly for your anatomy class, anatomy thing, but here less question is not so much important in physiology. So there are two venous system, deep superficial venous system and the deep venous system. Collateral circulation. This is important, particularly in abnormal condition. Normally, our coronary circulations, they don't have collaterals. Collateral means, suppose this is the septum. One septal branch is there. So this septum, this branch is to supply the septum. Now what happens? If this branch is blocked, the septum is going to suffer because it is not having additional supply. So this sort of circulation is called end arteries. End arteries. The artery, it will entering here by forming capillaries, it will end up here. And collaterals, another branch from other arteries may come to supply the same septum or some other part of the heart. So this branch is called collaterals. When this will be blocked, no problem, I will get blood from that collaterals. So when our heart is in crisis, they develop collaterals within few minutes. They develop for our survival. It is the strategy. It will try to make you survive. Okay. So this sort of things are there. And there are anastomoses also. Anastomosis what? Normally there is no anastomosis. Two types of anastomosis are there. One is anatomical anastomosis, another is physiological anastomosis. Anastomosis is one branch is joining with the capillaries of another branch. Okay. Now these are anatomic branches, but they may not be functional. They may not be functional. But when our body or our heart is in crisis, this anastomosis may become active. It only happens when there is an attack. When there is ischemia, gradual ischemia, then collaterals will develop. Then this anastomosis can develop. Okay, so this is always not there. These arteries are always end arteries. No anastomosis, no collaterals. But when there is need, it is needed, they will form collaterals, they can form angiogenesis. Means from other branch, there will be formation of capillaries from other branches, and these capillaries will be coming to this affected area to supply the blood. So these are usually after one attack, what is there? The doctors give Doctor gives 72 hours time, isn't it? Because within these 72 hours, collaterals may develop and angiogenesis may occur, more capillaries may develop to the injured area. So this angio become regenerated. The patient may get survived. And after that, if it is not developed, what will happen? These tissues will be rotten, and when other part is okay, when this will be contracting, there will be bulging, and ultimately blood will be coming out through this area. Huh?
Collaterals and anastomoses are the same thing or what?
No. Collaterals means, as I said, end arteries means one artery is producing capillaries and ending into this tissue. Collaterals means extra cardiac anastomosis. So during emergency, during when we need, the heart is suffering. Okay, there is infarction. So it tries to recover from it. So it will get supply from outside the heart. It may get supply from the esophagus, esophageal artery, bronchial artery. So from there, it can have circulation. See, thoracic artery, pulmonary artery. So blood vessels from there, phrenic arteries. From the plants may come and form anastomosis. Coronary artery dominance means this is the posterior side. What is this artery? What is there? The junction of both the ventricles. There is sulcus. So one artery, branch of the coronary artery passing through this. And normally it is the branch of this. Normally it is the branch of left coronary artery, left circumflex artery. It will descend through this way. Now, this artery may get supply from here, both. And it is present in what percentage? 10 to 20% of people, of the population. But in most cases, this artery is supplied by right coronary artery. And in some cases, this is supplied by what? The left coronary artery. It means this artery at the back, running in between both the ventricles, this artery may get supply from the right coronary artery, may get from the right coronary artery, may get from both. And those who are having both, they are lucky. If that branch is affected, this area will get blood from that part also. Okay, so this is called dominance. So your heart, that part of the, if it is mainly from the left coronary artery, that means you are dominant to, your dominant blood supply is left coronary artery. If it is by both, that is both are dominant. If it is supplied mainly by the right, then it is right. This is the dominance, meaning of the dominance. Okay? In short, in 20% individuals, myocardium is predominantly supplied by the left coronary artery, and in 50% individuals, it is by the right coronary artery, and remaining 30% by both. Is that clear to you? What do you mean by dominance? This is what? Left coronary artery, circumflex. So I have taken a longitudinal section to see how this artery giving off smaller arteries. So now this from here, smaller arteries are entering within the myocardium. And in the subendocardial region, these smaller arteries are joining together, forming a plexus. Here, no such vascular plexus is formed in between. Why in the subendocardial region? Why not in the myocardial region? Because this subendocardial region very much prone to myocardial ischemia. Very much prone to this area suffers mostly. What it suffers? During ventricular systole, less blood is supplied to this area. To compensate, so that to ensure this, so that the subendocardial region get adequate supply, so they form a plexus, so that this plexus, through this plexus, the area can blood from this. Moreover, this part, this is subendocardial region. You can see they don't have any vasculature. This is 1/10 millimeter, very thin, 1/10 of millimeter. They don't have any vasculature. Now question is, these are living tissue, so where from they will get their nutrition, get the oxygen? Since they are very thin and they are in direct contact with the blood of the cavity, from that blood, this layer of subendocardium, very thin layer of subendocardium, they can draw their nutrient and oxygen. Understood? So this is another important aspect. Special features of coronary circulation. And this subendocardial region, there is plexus, vascular plexus, and see how they are producing. Main arteries remain superficially, from their branches are entering within the myocardium and producing capillaries, giving oxygen and nutrients to the myometrium. And some branch is coming down to the subendocardial region and they join together, forming a plexus. There are two types of plexus. One is plexus means network. Network of what? If it is network of axons, nerve tissues, axons, we call it nerve plexus or neural plexus. This sort of plexus is there in our GI tract. In between, there are two longitudinal and circular layer of muscles. Meissner's plexus. So this sort of two plexuses are there. So these are neural plexus, but here it is not the neural plexus, it is the vascular plexus. Is that clear to you now? Fine. Okay, now we have gone through the anatomy, means how they originate, the coronary blood vessels, coronary arteries, how they originate and how they distribute to the myocardium. We learned it. Okay. Now we will discuss about some peculiarities of coronary circulation. You may expect this in your exam. Okay? What are the specialities of coronary circulation? What are the peculiarities of coronary circulation? Same question, same answer. There are 8, 9, 10 points. So you have to remember those 10 points. So before I give you explain these points to you, Already 1 hour gone. What is cardiac output? Who can say what is cardiac output? Anybody? What do you mean by cardiac output? Okay. Okay, no issue. You can tell. Okay, no issue. Do you know what is stroke volume? Okay, today I am telling you what is your group? A. So in the next class I will be asking you same thing. Heart, ventricle. So both are contracting simultaneously. Both are having certain amount of blood within their cavity. Now their contraction, when they will be contracting, this blood will be ejected into the aorta, into the circulation. So each contraction, certain amount of blood is ejected into the circulation. Is that clear? What is that amount? By one beat, each ventricle, how much blood it ejects into the circulation. That is called stroke volume. The volume of blood ejected by the ventricles, by each ventricle in each stroke. That is stroke volume. And its normal value is 70 milliliter. In echocardiography report, you will find SV, stroke volume, this mass. If it is 40 ml, means less. If it is 80, very good. 70, good. So normally it is 70, fine. So now you know it is normally, it may go beyond 80, 90, 100, 120 even. But it can come down also. When you will be doing exercise, heart is beating. So gradually what will happen? It will be increasing its stroke volume. More blood is coming into the heart, so more blood will be ejecting. So stroke volume, but there is a limit. Beyond that, it cannot do it. So normally up to a certain limit, stroke volume will be gradually increasing with when cardiac activity is increased. When cardiac activity? When we do muscular activity. So then only cardiac activity is increased. Fine. The myocardium is highly vascular. A lot of capillaries are there. Only why? To supply the blood and to supply the nutrients and to supply the oxygen. Highly vascular means they will get, this wall will get enormous circulation of blood. The blood flow should be more in comparison to other tissues. In 1 minute, every gram, what is the weight of heart? Okay, fine. 300 gram. 300 gram. If this 300 gram heart, they get 225 ml of blood in 1 minute. How much blood every gram will get? Got it. 300 gram. They get blood, how much? The total heart gets how much? 225 ml in each minute. I want to know how, 1 per gram of this heart muscle, how much it will get? Divided by 30. How much it comes? It will be coming to around 70 to 80 ml. Milliliter. If you divide it by 300. So its consumption, utilization or blood flow is, in comparison to other tissues, it is much high. 3 organs. One is heart, another is our brain and the muscles. They are the vital organs. We have seen, we have limited amount of blood. How much blood we have? In your body, how much blood you get? Are having any idea? Huh?
5 to 6 liters.
5 to 6 liters. And in respect to your weight, body weight, in some book it is written 1/11th of your body weight, 1/10th of your body weight. If your body weight is 100, 1/10th is 5, so 5 liter. This 5 liter of blood has to be circulated throughout my body in every minute. I have this limited amount, 5. This 5 liter has to be circulated. Now when this 5 liter is distributed, some part heart will be getting some part, this will be getting some part, muscle, intestine, skin. So all part will be getting a portion of this 5 liters in every minute. Now, and the muscles, they are vital organs. So I have started exercising. So my cardiac activity will be increased, muscular activity is increased. So they will be needing more blood. From where I will get this excess blood? I will curtail. I will curtail from others. Oh, my emergency is going on. So you will get, normally you used to get this much, now you will get less. Because now during this crisis period, digestion is not required. Absorption is not required. First I have to meet this emergency period. Circulation to the skin, I have to minimize. So in this way, I can cut the supply to other organs. I can minimize the supply to other organs. Now the extra blood I will be supplying to the vital organs. This is called redistribution of blood. Initially, normal condition, it is distributed like this. When it is emergency, then I redistributed it. Is this clear? I think you will remember this. So this is important. Now, this out of this 5 liter or 6 liters of cardiac output per minute, our heart uses 4 to 5% of the cardiac output. Means every minute. If it is 5 liter, 5% of 5 liter, 225. So it is consuming 225 ml of. How this 225 ml came? Value came? This way? Okay. So its metabolic activity is much more, since it has to contract continuously. Okay, without any pause, it has to function without any pause, no rest. So it has to be, everything has to be supplied immediately. Okay. Let us discuss the important aspects. First, peculiarities of coronary circulation. What are the peculiarities? The first point, it receives 4 to 5% of cardiac output, which is about 225 to 250 ml per minute. Or you can express it per 100 gram. In some books, it is given as per 100 gram. Simply 225 or 250 ml per minute. If you want to express it per 100 gram, so divided by 300, then it will be coming to, or divided by 3, because our weight is 300. If I divide it, then it will be coming to per 100 gram. And if you divide it by directly 300, then per gram. And that per gram, how much it will come? (250) or 250 divided by 300, 0.7 to 0.8 ml per gram. So this is the number one. Second is the blood flow to the left ventricular wall. Very important point. This point you have to write. Now tell me, this part of my body, it is getting blood through arteries. There is a pulse, we feel the pulse, artery. What? During systole, it rises. Then diastole again comes down. So when it rises, we can feel. So systole, diastole, I can feel it here. Because the blood is flowing in a phasic manner. Systole, blood is, you throw something on a pond, water. So when it will fall, it will produce waves, and these waves will be transmitting towards the periphery. Similarly, when blood enters into the aorta, it will produce a wave of systole. Now this wave will be transmitting through the arteries, and that you are actually there, you can feel. These are the pulse. Pulse of what? Pulse of blood that is coming because of systole. And when it is in diastole, heart is in diastole, no blood is coming. That's why it is, you can't feel the pulse. Again, when systole will be coming, this wave will be coming to here, you will feel another pulse. Clear? See, this is the wall. This is the wall. Now, the ventricular wall will be contracting. The muscles here, they will be contracting. As a result, what will happen? This layer will be squeezed. The layer will be squeezed during contraction. It has to, muscle will be contracted. When this muscle I contract, what is happening? It becomes stiff and contracted hard because of the squeezing. So during contraction, systole, this part will be squeezed. As a result, what will happen? This area will get less blood supply, less coronary flow, isn't it? If I compress it, then how blood can enter? Flow will be diminished, flow will be reduced. So when systole is there, systolic blood is entering through the coronary circulation. Since the wall is contracting, what will happen? Less blood will be flowing to the myocardium. This point is very important. Can you understand this? If you squeeze my hand like this, can blood enter there? Similarly, if the wall is squeezed because of the contraction of the myocardium, can blood vessels will be squeezed? No blood will be flowing, less blood will be flowing. So this is happening when the ventricle is in systole. So actually this is the one peculiarities different from other organs. During systole, particularly in the left ventricle, it has to generate how much pressure? 120, 130, 150 millimeter of mercury pressure. The muscle has to generate this much of pressure. So within this pressure, because of this pressure, the blood vessels will be narrowed. So less blood will be passing through during systole. But in other organs, during systole you will get more blood. During diastole you get less blood. But in case of ventricle, it will get more blood during diastole. Blood vessels will be dilated, so more blood will be entered. This is the peculiarities of coronary circulation, particularly for left ventricle. But right ventricle, it is also contracting. Its thickness is less. Wall thickness is less. Here wall thickness is more. And moreover, right ventricle has to generate less pressure, less force, only 25 millimeter of mercury pressure, very less. So they will not affect coronary blood flow during systole. Understood? So So you can see the coronary circulation in the ventricle or in the heart is biphasic. Meaning during systole flow will be reduced and during diastole flow will be increased. So increase, decrease, increase, decrease, increase, decrease. This sort of phasic blood flow will be there in left atrium particularly. This is another peculiarity. Number 2 point you understood? Number 3 point, already I explained. Subendocardial layer is compressed maximally during systole and therefore prone to suffer ischemia more than the other layers. Are you convinced with this? Number 3 points. During systole in left ventricle, if the middle part gets less, the subendocardial part will suffer most, and they are prone to ischemia infarction. That is the point. Subendocardial layer is compressed maximally during systole and therefore prone to suffer from ischemia and more than the other layers. That's why that has been developed. In the subendocardial plexus has been developed to counteract this. And the innermost layer, they are getting directly oxygen from the there for the survival. And number 4, oxygen consumption is very high, 70 to 80%. See, this point you have to understand. How much hemoglobin you are having in your 100 ml of blood? How much hemoglobin you are having in your 100 ml of blood? Okay. Let it be 14 gram %, means 14 gram per 100 ml. What is the function of hemoglobin? Carry oxygen. When it is fully saturated, every hemoglobin molecule can carry 4 molecules of oxygen when it is fully saturated. So in this condition, how much oxygen every gram of hemoglobin can carry? Their function is to carry oxygen. So 1 gram of hemoglobin, how much it can carry? 1.34 milliliter. Right, 1.3. Hemoglobin can carry, 1 gram of hemoglobin can carry, when fully saturated with oxygen, how much? 1.34 ml of oxygen. Okay? How much?
1.34.
34. So in your 100 ml blood, how much hemoglobin you are having? 14 gram. This 14 gram is present in 100 ml. Now I want to know how much oxygen is there in 100 ml blood. You are having 14 gram hemoglobin. So every gram can carry 1.34. So your 100 ml blood can carry can have 1.34 into 14. How much it comes? How much? Make it, you have, just for calculation problem, you have 15 gram of hemoglobin. So 15 into 1.34, multiply it. Means your 100 ml blood can carry 19 to 20 ml of oxygen. Per every 100 ml of your arterial blood carries 19 to 20 ml. Why 19? Why 20? 19 to 2. Why not 25? Why not 10? Yes, it will vary depending on the amount of hemoglobin. If your hemoglobin is less oxygen, that happens in anemia. In anemia, what is happening? You are having less hemoglobin. As a result, you can carry less oxygen to the tissues. So your tissue suffers oxygen deficiency. It will produce less energy. So you will feel lethargy, no energy. So low oxygen means no energy. Why? This oxygen will be utilized for generation of energy. Okay. So our blood, arterial blood, carries at least 19 to 20 ml of blood. Okay. So when this blood will be coming to my tissues, tissue will take up oxygen. So out of 100 ml, it takes up near about 5 ml of blood, 5 ml of oxygen. So 19 to take minus 5 ml taken by the tissues from every 100 ml. So the remaining amount of oxygen will be coming back through to the venous blood. So what will be the content of venous blood of oxygen? 19 minus 5, 14 ml. So your venous blood also having oxygen, and that is about 14 ml, 13 to 14 ml. How much it takes our tissue? From every 100 ml, it takes 100 ml of blood, it extracts 5 ml. Okay, but in our cardiac tissue, here if this arterial blood, when it enters in the myocardium extracts 80%, 70 to 80% of 20 ml. How much it comes? How much it comes? Near about 14 ml. So subtract it from 20 ml, you subtract this 14 ml, then in the venous blood, how much it will be there? So, see how it is power of extraction of oxygen from the arterial blood is much more than any other tissue. Now you can see it is how much activity it is performing, how much oxygen it is needing. So its oxygen demand is very high. When your cardiac activity will be more, it will need more oxygen. And the cardiac muscles are having one more thing, very important thing, myoglobin. And this myoglobin has got more affinity for oxygen than hemoglobin. Hemoglobin came with oxygen to the myocardium. So myocardial tissue is having myoglobin. Okay, leave this. It is mine. So it will extract as much as possible. So myoglobin will take up. MBO diffuse also low. There is within the cell there is low oxygen pressure. Within the blood there is high oxygen pressure. So there is pressure diffusion. There will be automatically diffusion. Other than this, so cardiac muscles, they are binding. They will bind extra oxygen with them. So when there will be deficiency of oxygen, it will supply this oxygen. So in that way, see how our system is developed, so that we don't suffer hypoxia. We don't suffer hypoxia. Hypoxia means we will be getting less energy. Our heart will not be pumping properly. So that sort of arrangement it has made. So oxygen consumption is very high. We have seen how it is being extracted. Here 70 to 80% of the AB difference is very high. In other tissue, AB difference is 19 ml and 14 ml. Difference is 5 ml. Whereas in cardiac tissue, difference is very high. 19 minus 13, how much it comes? 4 to 5 or 5, 6 ml. AB difference is very high. It means it is extracting more and more oxygen. Blood flow is regulated entirely by local. This 4 part, number point we will explain in other day. Next day, auto-regulation. Auto-regulation. At rest, number 6. At rest, where is number 5? Oh, 4 twice. So sympathetic address, it utilizes the energy from heart. Normally, under this condition, we use glycogen. But our heart muscle, most of the energy will derive from the glucose or glycogen. But heart most of the energy will derive from free fatty acids. That is another difference. It will be able to generate not only this, it can metabolize so many things. Starting lactic acid, pyruvic acid, this can metabolize. Normally, there is no communication between the larger coronary arteries, but many anastomoses exist between smaller arteries, we have seen. And next, degree of damage to heart muscle is determined to a great extent by degree of collateral circulation that has already developed. That is, at a portion got ischemia, or ischemia means vessel narrowing. There will be narrowing of the blood vessel. So less blood will be coming, less oxygen. So our heart will be producing metabolites, and this metabolite will create pain sensation. This pain sensation will be transmitted. So this is called myocardial angina pectoris. This pain is called angina pectoris. When there is angina pectoris, it means our cardiac muscle is getting less circulation of blood, getting less oxygen. When a particular artery will be blocked, then a particular area will be devoid of circulation. So it will undergo inactivities. So this is called infarction. This will be rotten, this area. Unless it gets collaterals from other sources. Otherwise, this area will die. This is called infarction. So depends on the last point. Degree of damage of heart muscle is determined to a great extent by degree of collateral circulation that has already developed or that can develop within minutes after occlusion. Get a photograph of this. All the points you have to remember. In your unit examination, in your one examination will be there after the ending of the series. So this question may be there. What are the peculiarities of coronary circulation? What is autoregulation? Explain the autoregulation of coronary blood flow. So that part I will explain later. Okay, now I am ending this. Previous class, previous experiment, we learned how to prepare nerve muscle, gastrocnemius muscle, sciatic nerve preparation. We know how to make a setup to demonstrate if we give a proper stimulus to the nerve, sciatic nerve, the muscle will respond by producing contraction. That we want to record it. One of the muscle, both are excitable tissues. Both are excitable tissues. It means if you give a proper stimulus either to the muscle or to the nerve, they will respond. How the nerve will respond? They will produce nerve impulse. And this nerve impulse will be transmitted, will be conducted along the nerve fiber, and it will go where? To the nerve endings. It will go to the nerve endings. Now if it is the nerve ending, it supplies a muscle. Now this impulse cannot go enter directly into the muscle. Because there is a junction in between, there is a gap in between. This impulse has to cross this gap, this neuromuscular gap, we call neuromuscular junction. So what is there at the end of this nerve? There is a swelling. We call it synaptic knob or button, synaptic button. So these are having, these nerves are, we told, there are different kinds of nerves in our body. We give different type of nomenclature. Depending on we divide them into two, medulated, non-medulated, myelinated, non-myelinated. Okay, if the nerves are having myelin sheath, then we call it myelinated, if not, non-myelinated. Similarly, we can divide, classify them depending on the neurotransmitter secreted from the nerve endings. So there are different type of secretory substances. So what are these? Acetylcholine. So these nerve will be those who secrete acid, they will be cholinergic nerve. Those who will be liberating adrenaline, they will be adrenergic nerve. Those who will be secreting serotonin, they will be serotonergic nerve. This way you can classify according to type of neurosecretions. They secrete from the nerve endings. Okay, we also classify them depending on the nerve carrying impulse, whether it carries from the periphery to the center or it carries impulse from the center to the periphery. So if it carries impulses from the periphery to the center, we call it sensory nerve or afferent nerve. And if it carries instruction or information or impulses from the center to the periphery, we call it motor nerve or efferent nerve. Okay, in that way also we classify. We also classify nerves into based on their diameter and based on the velocity of nerve impulses. And this part is important for, this type of classification is important for your exam. So you learn it properly. And this nerve, sciatic nerve, it is a modulated nerve, and from its nerve endings acetylcholine is secreted. So when it is stimulated, it will be responded by producing action potential, means nerve impulse. So nerve impulse will be coming to the neuromuscular junction. From the neuromuscular junction, acetylcholine will be liberated. Now this acetylcholine will bind to the sarcolemma. So it means postsynaptic membrane. So means motor end plate. So they are having, this membrane is having receptor for binding acetylcholine. So this acetylcholine, liberated acetylcholine, will bind with the receptor, and binding of this receptor with this receptor, acetylcholine, when binds to the receptor, then what will happen? Some channels will be opened, sodium channels will be opened. So sodium ion will be entering from the ECF, means synaptic cleft. In the synaptic there are ECF. From this ECF, sodium ion will be entering within the sarcoplasm. So the muscle cells are also excitable cells. They are also having resting membrane potential, means inside is negatively charged, outside is positively charged. Now if from outside, means ECF, sodium enters, inside will gradually become less negative, and when more and more sodium ion will it will be depolarized. When it will exceed the threshold level, then action potential will be developed, and this action potential will be traveling along the sarcolemma. Now the sarcolemma is entering within the muscle fiber, within the muscle cell through T-tubule. Now through T-tubule, then what will be there? There will be cistern. From the cistern, there will be liberation of calcium ion. Now calcium ion will initiate EC coupling. Excitation, contraction. So because of excitation, calcium is liberated. Now this calcium ion will initiate the process of muscle contraction. So this is called EC coupling. So this requires time. One is traveling time stimulation, at a part of the stimulation. From there it has to come to the neuromuscular junction. Then crossing the neuromuscular junction, it may take some time. Then liberation of, means passage of the impulse through the sarcolemma into the T-tubules and means to the sarcotubular system. Then liberation of calcium ion. Now then calcium ion will initiate sliding filament, means sliding of actin and myosin, means muscle contraction. Okay, so these are the different phases. Okay, so when suppose this is the nerve. So here is the electrode. Through this electrode, I gave stimulation to the nerve. তাহলে আমরা আগের দিন দেখেছি কি, we saw when we gave stimulus to the nerve, then muscles contracted by responded by making a twitch. That we have recorded, and this record is called simple muscle contraction. And we have also seen immediately after giving the stimulation, the muscle did not respond. It responded after a certain time. It took some time. That time is called latent period. Why this latent period? Explain. Why this latent period?
So the time taken to
Travel through the.
local and ultimately calcium will.
So one time you will be for the passage of impulse to the neuromuscular junction. Then some time will be required for traveling the junction. Then some time will be required for traveling through the circuitubular system to the cistern. Then it will produce calcium, and then there will be some time, there will be some metabolic reactions. Through these reactions, ATP will be produced, and this ATP will be required during sliding of actin and myosin. So they will produce some time. So this time is required for traveling, crossing the neuromuscular junction, and initiating excitation, contraction, coupling, and contraction. Okay. Beside this, another factor is there, because no weight. So I will be contracting. If I place a 5 kilo weight, then if I have been asked to contract, then it will take longer time to initiate the contraction. Why? Because of inertia of this weight. This is, so another cause, this lever, short arm, long arm, these are the resistance. It has to, first it has to overcome this resistance, then it will be able to contract. It is the inertia of this. Okay, so these are the cause of latency. Okay, so then there will be contraction. So we know in a simple muscle contraction there are 3 periods. One is contraction period, latent period. Second one is contraction period. Third one is, today we will be learning what? Whether temperature has got any effect on simple muscle contraction. Whether temperature, it may be hot, it may be cold. So we'll be applying, we know we used one fluid, that is ringer fluid. We know the composition. So instead of normal ringer fluid of normal temperature, we'll be using hot, say of 40 degrees centigrade. We will not apply more than 40 degree, 42 degree, 45. Why? Because the enzymes are very sensitive to temperature. First, their action will be increased, their activity will be increased. Thereafter, their action will be decreased. Why? There will be denaturation of the proteins. Denaturation, the enzymes are protein in nature, so the enzymes will be denatured. This actin and myosin, they will be denatured. As a result, actin myosin will not contract, will not slide over each other, and the enzymes will not produce any energy. Because enzymes are also protein, they will also be damaged at high temperature. Similarly, at low temperature also same thing can happen. Okay, so let's see. So we'll not give that much of temperature at a reasonable increase, say from 28 degrees centigrade to 40 degrees centigrade, 40 degrees centigrade or 38 degrees centigrade. Let's see what happens. What happens to latent period? What happens to contraction period? What happens to relaxation period? Then we will apply cold saline, cold ringer. Let's see what happens to latent period, contraction period, relaxation period, and height of contraction. These things we want to see the results. Okay. what our theory says. What our theory says. We learn theoretically velocity of nerve impulse is increased when there is increase in temperature. Means velocity of ambulance depends on temperature. Higher the temperature, higher will be the velocity. That too up to a limit. And lower the temperature, lower will be the velocity. Okay, so this theory we are having. Another theory knowledge we are having, that is, the muscles are having protein, and these proteins are having viscosity. The sarcoplasm having viscosity. So again, viscosity is also affected by temperature. With temperature, viscosity falls. With reduction in temperature, viscosity rises. When we reduce the temperature, viscosity increases. Viscosity is a resistance to contraction. So, increase in viscosity with low temperature, there will be increased resistance. So, height of contraction will be less. It will be able to generate less force. And the reverse, with increased temperature, viscosity will be less. So, less resistance easily can be contracted. So, height of contraction will be, it can develop more force. Okay. So, one thing learned, it increases the velocity of nerve impulse. Another thing, it reduces or increases the viscosity. And temperature increases the metabolic process. We know enzyme activity depends on pH as well as temperature. Important factor. At a particular temperature, that is called optimum temperature, at that temperature, the enzyme activity is maximum. And if you go beyond this level of temperature, the activity will gradually become less. So, if we increase the temperature, this activity will be rising. Metabolic activity will be, why metabolic? Because all the in muscle contraction, we have to generate ATP. We will be generating ATP by different biochemical processes, biochemical paths. One is glycolysis, TCA cycle, and oxidative phosphorylation and beta oxidation. Okay, all are enzyme guided. So, if I increase the temperature, activity of these all enzymes will be increased. So, there will be more production of ATP. Now, ATP will be available readily to the muscle. So, with that, it can contract easily. But with lower the temperature, all the activity will be, enzymes activity will be less. So, there will be less ATP will be available to them. So, it will be able to generate less ATP. Okay. Now, you can understand. Now when we apply the hot saline, when we apply the hot ringer, what will happen to the latent period? Whether latent period will remain same, or it will be increased, or it will be decreased? Latent period will be decreased or increased. Why decreased? Because traveling time will be less. Now it will travel faster. So, there will be, with temperature, higher the temperature, lesser will be the latent period. Very good. Okay. Then, height of contraction period will be less, isn't it? Because why? Viscosity is less. And force of contraction is more. Why? More ATP is available, viscosity is less. It gets more ATP for muscle contraction and less resistance because of lowering of viscosity. Okay, so these are the causes. And relaxation, viscosity less, it will take less relaxation time. So what will be your observation with hot saline? Reduction in latent period, reduction in contraction period, reduction in relaxation period. Now, if I give cold saline, what will be our observation? Reverse. Latent period will be increased. Now it will take more time, more troubling time. Velocity will be decreased, and contraction, height of contraction will be reduced because it has become more viscous, and enzyme activity is lessened. Now less, so more available ATP is less. So what will happen? Contraction period will be longer, but height will be less. Relaxation period will be longer because viscosity is now more. Let us see. So point cover is
viscosity or enzyme activity.
So this is the normal curve. This is the first curve at room temperature. Green one. Here we have given the stimulation. So for the first curve, what is the latent period? C1. What is this? C. Na B, B, A, B, C, D. A to B or A to C or A to D. For normal curve, A to C. Correct. A to C. Now, how can you determine the latent period? You have to calculate how many frequencies are there under this A to C segment. How can you do it? Okay. Starting point here and ending point. So there is one cycle plus this was, it is 0.25, 1/4. Then how many frequencies are there? 1.25. Isn't it? So 1.25 into 0.01. This is the latent period. And contraction period. So you have to draw a vertical line from the tip, maximum height of contraction. So this will be the contraction period and this will be the relaxation period. How many frequencies are there? Count it. Change your glass. What happened to the latent period? Whether it is reduced or increased? Decreased? Means why it is reduced? It did decrease because the velocity is now faster. That is the reason. Okay, contraction period reduced or increased? Reduced. Why it is reduced? Viscosity, decreased viscosity and increased supply of oxygen, or not oxygen, ATP, increased metabolic activity. Okay, and relaxation time is reduced. Why relaxation time reduced? Same for the purpose of viscosity. For the purpose of viscosity. Okay, now the blue lines. What happened to latent period? Increased. Why? Fine. And contraction period? Increased. Why? Viscosity increased, metabolic supply of ATP is reduced because of low metabolic activity of the enzymes, and relaxation period again because of increased viscosity. And ATP also ওখানে ATP, unless ATP becomes bound, this the bridge that has been formed, this myosin head will not be detached. When ATP binds to the myosin head, then only it is detached. So since it is remains attached to there because of lack of ATP supply, so relaxation process is prolonged.
As you have seen in the earlier class, we have seen the effect of the muscle twitch in normal temperature. Now we will, in the similar way, we will give a stimulus and we will be using a hot temperature. So how will we achieve the temperature? We will either heat the solution, the linear solution, or we will cool it. So for cold temperature, it will be around 10 degrees centigrade. And for hot temperature, it should be around 40 degrees centigrade. And it should not exceed 42 degrees centigrade. So suppose we are taking it as a hot drinker solution. So this temperature will be around 40 degrees. So someone press the key and give the stimulus. So is there any change from the earlier class what you have seen in normal temperature, in the stimulus and in the movement of twitch of the muscle? Is there any change? What change you could notice? Okay, there is difference in height of contraction. This height is definitely increased, and this curve is narrower. The contraction is beginning early, and there is also early relaxation in this case. And you can see that here the latent period is also less. It is decreased. Like in normal ringer, you can see that this one is the latent period. There is some difference in the time period. So sir has already told you that why these things are happening. There is increase of enzymatic activity. There is reduced viscosity of the muscle. So these things contribute in the early contraction or early twitch of the muscle and in the decreased latent period. So this one is the theory. Some one of you take a picture of the theory. Done.